Hello everyone, welcome to this video on power and sample size estimation. Power and sample size estimations are measures of how many pages are needed in the study. They are an important requirement for grant application and research publications. So what type of study should have a power calculation? Well, the answer to this question is almost all quantitative studies are eligible for sample size estimation. When researchers recruit too few participants, the study becomes underpowered and prone for both false positives and false negatives. This leads to inconclusive results, waste of time, money, and manpower. Similarly, when the study is overpowered, that is having more participants than that is actually required, there's waste of resources. So the most frequent question asked to statisticians is what exactly is the sample size required? Often clinical researchers outsource or pass on this buck on sample size calculation to a statistician. But statisticians are not themselves medical persons. So the expertise of the clinical researcher is often required in formulating the appropriate sample size for the desired outcomes. In this slide, I'm presenting a 5-step approach to determine the power and sample size. The first step is to define the outcome variable of interest and comparison group, and then formulate a hypothesis. The outcome of interest may be a nominal, categorical, or scale variable. The comparator may be between groups or matched pairs. Now we need to formulate a hypothesis. So what is a hypothesis testing? A hypothesis is an idea that can be tested. This is the simplest explanation that can be used, although this is not a definition by itself. There are two different terminologies that need to be understood when it comes to hypothesis testing. One is a null hypothesis, and the other is an alternative hypothesis. A null hypothesis can be regarded as a status quo, or you can call it existing state, or it may well be a widely held belief. When you challenge the status quo, it becomes an alternative hypothesis. Let me give you a dental example in this regard. We all know that adults with periodontal disease have more number of anaerobic bacteria in the oral cavity, and this is normally not the case with children. So what about children with the habitual nail-biting habit? Do they express higher anaerobic bacterial count compared to those with no nail-biting habit? The logic for this argument stems from the assumption that autoinoculation of environmental microorganisms, compounded with less than optimal hand hygiene in children, can change the bacterial microflora in those with chronic nail-biting habit. So the null hypothesis would be that there is no difference in the anaerobic bacterial count in those with or without nail-biting habit. And let's say I want to challenge this null hypothesis and test if anaerobic bacterial count is present more in those with chronic nail-biting habit. So in that case, my alternative hypothesis is that there is a difference in the anaerobic bacterial count. There are many such examples, and the one that interests me the most is the bidirectional relationship between periodontal disease and diabetes. So let's go to step 2. Right, in this step we need to determine the effect size. The effect size can be a small, medium, or large one. To detect small but clinically relevant change, a large sample size is required. Often small effect size is used as a gold standard, but this is easier said than done because of the difficulty in recruiting participants with similar characteristics in the test and control group. While deciding on the effect size, what constitutes a meaningful difference needs to be established. This can be determined from previous studies, so literature search can be a good starting point for this. In this stage, the desired power can also be determined. Imagine a study that compares two different antihypertensives. Even if the study fails to identify any clinically meaningful effect size, it may well be that the newer drug can be a cheap alternative to the existing one. As such, it is simply not correct to disregard if the findings do not show any acceptable effect change. So the third step is we need to determine the correct statistical test to use for the power calculation. And this again depends on the nature of the data, which can be either nominal, ordinal, or scale variable as discussed earlier. The statistical test that is appropriate is determined by the type of exposure and primary outcome variable. So the fourth step is to perform the calculation, and the appropriate sample size calculation is performed with the help of software. There's loads of software, there's so many paid ones and a lot of free ones as well. The popular among the paid ones are OS, BSS, you have NetCalc, and then you have Minitab. There's equal number of free ones which you can download from the internet. GPower is one among the popular ones. Then you have SAS, Stata. I'm not sure if SAS and Stata are free. I haven't used those anyway. So it could be one of those. There's many more. There's loads of them here. So you could use the software to perform a sample size calculation, and then comes the final step. Oh, before we could move on to the final step, and when you're doing the statistical calculation, and if you're not happy, then you have to go back to step 2, and you could try with a different effect size, and you just have to keep you know changing things. I mean, fine tune and see whatever that comes as a desirable option for you with the available resources. And the final one would be, so you got a state and grant and manuscript. So finally, you write the formal sample size statement for the such grant application or the manuscript. The formal statement will include the effect size, then your power analysis, the alpha preset values of whatever alpha you want to keep. Often is 95% or 0.05, and the statistical test or statistics considered and the sample size. So all this goes in, and that is all it is. I mean, the 5-step approach. 2x2 box here. 2 of them are shaded in green with a smiley face in it. In the top one, you have the null hypothesis, which is true, as such the alternative hypothesis is rejected. Similarly, in the bottom one, the alternative hypothesis is true, and so the null hypothesis is rejected. This part is pretty straightforward here. The problem comes with the false positives, or type 1, and false negatives, type 2 error. So I'll focus on reducing the type 2 error here. So we're looking at the power. So power is the probability of rejecting the null hypothesis when the alternative hypothesis is true. Increasing the power will reduce the probability of type 2 error. So what we can say is power is directly proportional to sample size, but inversely proportional to type 2 error. Hence, increasing the sample size or power will invariably reduce the type 2 error. I've done a sample one for you here. I mean, a common expression of power is 1 minus beta error probability, and that can be seen here. If you have a power of, let's say, 80%, and in decimals that would mean 0.8. So the type 2 error is reduced to 20%, or you can call it in decimals. You call it as 0.2. So if I'm going to keep a power of 90%, then my type 2 error is actually reduced to 10% or 0.1. Very similar to the table presented for hypothesis testing in the earlier slide is the one for screening and diagnostic accuracy studies. Here the 2x2 table used for predicting the sensitivity, specificity, then the positive predictive values and the negative predictive values. This is a very, very common one, very similar in all respect to the hypothesis testing 2x2 table. Right, so we got all the ingredients necessary to do the sample size calculation. So what I'm going to do now is to open this software called GPower, which is a freely available software on the internet you could download. There's many other things. You could get a paid one or the free ones. The paid options would be like SPSS, MedCalc, Minitab. They all have this sample size calculation and power analysis. GPower is a fantastic one. It's freely available, and there's many other things I'm not familiar with. You could do in Stata, SAS, and many other things here. Right now, I'm going to do a t-test now. A t-test would, well, example would be, let's say I'm looking at a scale variable, and assuming that we are trying to test a new drug for hypertension. Let's assume we're testing a beta blocker to a new calcium channel blocker, or an ARB, or an AC inhibitor. So we're going to look at the mean reduction in the systole or diastole. So we need to calculate the sample size for that. So we're doing the t-test. So the statistical test I'm going to do is the family comes in the t-test family, and I'm going to look for the mean difference between the two groups. So I'm going to click on that, and the type of power analysis I'm going to do is I'm going to do a priori. The reason why I choose a priori, because normally this is what you do before you start on the project or research. You do the a priori, and provided you have all the other preset variables which you have got a unified consensus on, for example, the alpha, the beta, and the effect size. So then I'm going to try the two-tail. The reason why I tried two-tail is because I'm not sure that I could get the results either way. I mean, the results could be that the new drug is probably not reducing the systole or diastole, and it may even worse. Actually, it's probably increasing the blood pressure. So I'm going to try a two-tail. I mean, unless otherwise, if the investigator is familiar with the situation and is absolutely confident that the new drug is better to the older one, then he could still he could still go for a one-tailed. And one-tailed is sometimes better if the investigator take the risk because it has a higher power. A higher power would essentially mean a larger sample size. Yeah. But again, a two-tail will have a more sample because you're doing on both direction, or either it could go less or higher. So, and the next one I'm going to do is the effect size. I'm going to keep the effect size here as 0.2. 0.2 is a small effect size because I want to capture the smallest meaningful difference that can be attained here. So that's what I want to calculate. So keeping a small effect size. A small effect size also essentially would mean that you need to have recruited more larger samples. So let's see with alpha. Alpha is the false positive, so I'm going to keep that as 5%. The decimal of 5% is 0.05, so I'm keeping that. And what that essentially means is there's a 1 in 20 chance of you're going wrong. So that's why you keep an alpha of 0.5 or 5% or 0.05. And the power I'm keeping, I mean, there's always a thing that you always keep the power as 80. So I'm keeping it 80 and above, not less than 80. So the desired, the widely accepted one is 80 and above. So 80 at times people keep it at 90. And the location ratio, I'm keeping that as 1. I mean, that's the assumption I had in mind because for the sake of simplicity, so I'm keeping as 1, which essentially means is that the number of people in sample size, sample group A are same as group B, or group 1 the same as group 2. Right, now I key in all the details now, so I'm going to just click on the calculate. Right, and that's the number I've received here. I've got 788 samples. So because I kept my allocation ratio as 1, so that means I got equal number of participants in group 1 and group 2, so it's 394 and 394. Right, now let's do a univariate analysis, and I'm going to change, instead of two tail, I'm going to keep it one tail. Although one tail is more robust, but it's still, as I said, it measures only one in one direction. That is, if the researcher is absolutely confident that the new drug is definitely going to reduce the systole or diastole for certain, then you could go in for a one tail. Yeah, I mean, these are all just experimenting. There's no fast and hard rules that they got to stick on with it, because you're still not, you haven't submitted your project proposal to the grant, yeah. So you could experiment all that now. So I'm going to try that, and I got a sample size of 620. Right, I'm going to do a multivariate analysis. So I've kept my tail as one tail, and I'm going to adjust my power here. So I'm going to keep my power to 90. So what that means is, normally if you increase the power, you're going to essentially increase the sample size as well. So you got more number of samples. So I got 858 samples now. So if I'm going to keep a two tail, my sample is going to be even more. It's going to end up more than 1,000. Right, now I got 1,054 samples. So if I keep a two tail, right, now all these details, I can just store it in my, all this information, what I did, it's all here. So I could click on the print option, and I don't really have to print it always. I could still save it as a PDF file. So I'm keeping it as a PDF file, and I could mail my people, the study collaborators, and the project funders. I could mail them. Replays, yeah, right. So that is it. Yeah, do you have any questions at all? Well, I'm sure you're going to have a lot of questions, because we just started off from middle now. Right, so that's the end of the lecture. Hope this video is of some help to you. If you have any difficulty in understanding, do not hesitate to contact me. Thank you very much. Enjoy your day and have fun.